Hello and welcome back to my channel, guys. And today we are going to discuss our first kingdom, that is Monera. In the previous videos we have discussed the introductory part of biological classification, and then we have discussed the five kingdom classification proposed by R.H. Whittaker, and on what basis, on what characters he classified the kingdoms into five. So today we are going to take up the first kingdom, okay, and this is Monera, okay, or this includes prokaryotic unicellular organisms, unicellular organisms, okay. And if I say in broad terms, we are going to take Monera into two categories or two groups. We are going to discuss in two groups, that is Archaebacteria and Eubacteria. And in Eubacteria we are going to again classify them into two, that is bacteria and cyanobacteria, or you can call it as a blue green algae, BGA. Okay, this is the broad concept or broad map of what we are going to discuss, okay, in Monera, kingdom Monera. So starting with the normal introduction of Monera. So Monera consists of bacteria, and bacteria are the biggest member of this kingdom, okay. Bacteria are the biggest members of this kingdom, okay. The next one is this kingdom includes bacteria, cyanobacteria, mycoplasmas, actinomycetes and rickettsia, okay. So this includes bacteria, cyanobacteria, mycoplasmas, actinomycetes and rickettsia, okay. These are the organisms that are being included in Monera, okay. So let's start with the Archaebacteria, okay. Let me clear this first. Archaebacteria, okay. So what is Archaebacteria is, we are going to discuss this now, and also the importance of Archaebacteria as well, and what are the main characters of the Archaebacteria. Yeah, so this group is the most primitive group, okay. Most primitive prokaryotes are included in Archaebacteria. So first point is most primitive prokaryotes are the Archaebacteria, okay. In this, the cell wall is made up of proteins and non-celluloic polysaccharides. Cell wall is present and made up of protein and non-celluloic polysaccharides. Okay, clear? Most primitive, and cell wall is made up of protein and non-celluloid polysaccharides. Clear? Then the next point is cell membrane is present, and cell membrane is, or say cell membrane has glycerol and other lipids. We need to remember this, that cell wall is made up of what and cell membrane constitute what, because these are the 1 mark questions in your in question papers. Okay, so do remember these points. Now, why this group is so unique? Because they have a RNA which is 16S RNA, which make this group unique. Okay, 16S RNA makes them unique and helps in placing them in separate domain, called archaea. Okay, 16S RNA. Clear? Next point is their habitats. They are found in extreme habitat. Okay, so extreme habitats like they are found in like hot sulfur springs, or say deep sea and salt marshes, etc. These are the extremities conditions of our earth, and they are present in these also. Okay, and Archaebacteria are called as living fossils. Why? Because they represent the earliest form of life on earth, and they are still present, so they are known as the living fossils. Okay, so who are known as the living fossils? Archaebacteria are known as the living fossils because they represents the earliest form of life on earth. Okay, I hope this point is clear. So let's discuss the types of Archaebacteria now. Types of Archaebacteria. There are 3 types of Archaebacteria. Number 1 is methanogens. Okay, so what are methanogens? They are obligate anaerobic form of bacteria. They are obligate anaerobic form of bacteria, and they are present in marshy areas. Present in marshy areas, and they form methane. Okay, which is an important gas of biogas. Okay, and if I take some examples of methanogen, let it be methanobacterium, and another one is let it be methanococcus. Okay, these two are the examples of methanogens. There are many more, but we are mentioning only two. Okay, so number second is halophiles. Okay, halophiles are, these are also present in extreme salt condition, found in extreme salt conditions. Clear? And they can trap solar energy for what? To produce ATP. Clear? So examples of this halophiles are Halobacterium. Okay, let's take the last but not the least. Third one is thermoacidophiles. Thermoacidophiles. And what are the basic character of thermoacidophiles? They are aerobic bacteria. Okay, and they are found in hot acidic sulfur spring. Found in hot acidic sulfur spring. Okay, and they can, oxidize sulfur to sulfuric acid, H2SO4. Okay, and these, the examples are sulfolobus. Okay, let's take another one, that is thermoproteus. Okay, thermoproteus. Clear? Now you can actually relate them. Methanogens produce methane. Okay, halophiles present in salt condition and can trap solar energy, which is used to produce ATPs. Okay, ATP is what? Energy. Okay, this is the type of energy which the living cells utilize to survive. Okay, to proceed the biochemicals reactions, or say metabolic reactions. Now thermoacidophiles, these are they are present in hot acidic sulfur springs, and they are doing what? They are oxidizing sulfur into sulfuric acid. Okay, so up to here I guess everything is clear. Types of Archaebacteria and the main characters of Archaebacteria. Clear? So let's move to the last point of this video, or this topic, Archaebacteria, that is the importance. What is the importance of Archaebacteria? Every organism in the earth is present because of some reason, and some work is to be done by them. So Archaebacteria are also here, and these are used in modern biotechnology to produce some productive products too, which are useful for humankind. Then also methanogens are used to generate biogases. So generation of biogas. Okay, Next one is thermophilic enzyme. Wait. Thermophilic enzymes from thermoacidophiles. Clear? Next, biosensors are also being produced or made by these Archaebacteria, and the restriction enzyme, etc. Okay, these are some important points, or say importance of Archaebacteria in the living world. Clear? So Archaebacteria are the part of Monera, kingdom Monera. Kingdom Monera is prokaryotic unicellular organisms, and bacteria are the biggest member of this kingdom. Okay, and we have seen that Monera is divided into two divisions, that is Archaebacteria and Eubacteria on their evolutionary achievements. And further, the eubacteria is being divided into two, that is bacteria and cyanobacteria. Cyanobacteria is also known as the blue green algae, that is BGA. Okay, so we have discussed here the first division, that is Archaebacteria. Okay, Archaebacteria is the most primitive organism in the living world, which is also, this is why this is known as the living fossil as well. And the cell wall of Archaebacteria is made up of proteins and non-cellulosic polysaccharides, and whereas their cell membrane is made up of glycerol and other lipids as well. Clear? And we have already discussed that there is 16S RNA which actually make them unique, and because of that they have put these are being placed in a separate domain which is known as archaea. Clear? And after that we have discussed that they are found, their habitat is that they are present in the most extreme conditions, like hot sulfur springs, salt marshes, and salt marshes actually, sorry, and deep sea. These are some places and most extreme conditions where no living thing can survive, but these Archaebacteria can be found there. Okay, so and they are classified in three types. Archaebacteria are classified in three types, and those three types are methanogens, halophiles, and thermoacidophiles. Methanogens are what? They are the anaerobic form of bacteria, and present in marshy areas, and they produce methane, which is important for important gas of important for biogas. Okay, here generation of biogas importance we have discussed here. Okay, next one is halophiles. They are extremely anaerobic bacteria found in strong salt conditions, and they can trap solar energy to produce ATP, which are further used by the living cell as well for their survival. Okay, next comes the thermoacidophiles. So thermoacidophiles are what? They are found in the hot sulfur springs, and these bacteria can transform, or say can oxidize sulfur into sulfuric acid. Okay, so these are the this is the part of Archaebacteria we have discussed, and in the next video we are going to start with Eubacteria, the second division of Monera, kingdom Monera. Okay, so I hope everything is being clear till now. So be in link and watch the previous videos as well, and do subscribe, like and share my channel, my video. And if you have any doubt, please write it down in the comment section, or send me a mail. I'm providing you my mail ID. You can contact me there as well. and if you need any tuition relating query, you can mail me there. Okay, so thank you and stay safe.